What if the cure for every disease already exists within your body? Imagine a world where cancer becomes a manageable condition, where emerging pandemics are stopped before they begin, and where the common cold is relegated to history books. This isn't science fiction. It's the tantalizing frontier of immune reprogramming, where scientists are learning to harness and redirect our body's natural defense systems to fight virtually any disease with unprecedented precision and power. Throughout human history, disease has been our constant companion and greatest adversary. From ancient plagues that decimated civilizations to modern pandemics that bring societies to a standstill, from cancers that strike indiscriminately to genetic disorders that follow family lines for generations. Illness has shaped our lives, our cultures, and even our evolution. Yet for most of this history we've been fighting with blunt tools, herbs, chemicals, surgeries, and prayers. But in the last few decades, something remarkable has happened. We've begun to understand that the most sophisticated disease-fighting system isn't found in pharmaceutical labs or hospitals, it's already inside us. The human immune system has evolved over millions of years into an incredibly complex network of cells, proteins, and signaling pathways capable of identifying and eliminating countless threats. It remembers past infections, learns from each encounter, and can deploy highly specialized responses to almost any pathogen it faces. The challenge has always been control. Our immune systems sometimes miss threats like cancer or certain viruses, attack our own tissues in autoimmune disorders, or trigger excessive inflammatory responses that cause more harm than good. What if we could change that? What if we could reprogram our immune systems, like software updates for biological hardware, to recognize and eliminate any disease with perfect accuracy and without side effects? In this video, we'll explore the cutting-edge science of immune reprogramming, from the groundbreaking CAR T-cell therapies already saving lives to speculative future technologies that could transform medicine completely. We'll examine how scientists are learning to edit, enhance, and redirect immune cells to target diseases that have long evaded natural immune responses. And we'll consider the profound implications this technology could have for human health, longevity, and our relationship with disease itself. In 1995, a researcher named Ralph Steinman was diagnosed with pancreatic cancer. As a scientist who had spent decades studying the immune system and discovering key cells that regulate immune responses, he faced a cruel irony. The very system he had dedicated his life to understanding was failing to recognize and fight his own cancer. Steinman survived long enough to be awarded the Nobel Prize for his discoveries, but died just days before the announcement. His story highlights both the remarkable potential and the current limitations of our natural defense system. To understand the revolutionary potential of immune reprogramming, we first need to appreciate the extraordinary complexity and capability of the immune system in its natural state. Composed of trillions of cells and dozens of specialized cell types, the immune system is less like a single organ and more like a sophisticated military organization with intelligence gathering, communication networks, specialized weapons, and memory systems. At its core, the immune system performs one critical function: distinguishing self from non-self, and eliminating anything identified as foreign or dangerous. This seemingly simple task involves multiple layers of defense and remarkable specificity. The first line consists of physical barriers like skin and mucous membranes, followed by innate immune responses that react quickly but broadly to common patterns found in pathogens. The adaptive immune system provides the third and most sophisticated layer, creating customized responses to specific threats and forming immunological memory that allows faster responses to repeat encounters. The key players in the adaptive immune system are B cells and T cells, two types of white blood cells with different but complementary functions. B cells produce antibodies, Y-shaped proteins that can bind to specific molecules, antigens, on pathogens, marking them for destruction or neutralizing them directly. T cells come in several varieties with different roles. Helper T cells coordinate immune responses. Cytotoxic T cells directly kill infected or cancerous cells. Regulatory T cells prevent excessive immune reactions. What makes these cells so powerful is their ability to generate incredible diversity. Through a genetic recombination process that occurs during cell development, our bodies can produce B and T cells with receptors capable of recognizing virtually any possible pathogen, even those the human species has never encountered before. It's estimated that our immune system can generate more than 10 to the power of 15, that's a quadrillion, different antibodies, a number far exceeding the total number of pathogens that exist. This vast recognition capacity comes with a challenge. The system must avoid attacking the body's own tissues. Multiple checkpoints ensure self-tolerance, eliminating or disabling immune cells that react too strongly to the body's own molecules. When these tolerance mechanisms fail, autoimmune diseases like rheumatoid arthritis, multiple sclerosis, or type 1 diabetes can develop. The immune system also maintains a delicate balance between activity and restraint. Immune responses generate inflammation, a necessary process for fighting infection but potentially harmful if excessive or prolonged. Regulatory mechanisms ensure that immune reactions are proportional to threats and resolve appropriately once threats are eliminated. Despite its sophistication, the natural immune system has significant limitations. Cancer cells often evolve ways to evade immune detection by downregulating markers that would identify them as abnormal or by creating immunosuppressive environments. Certain viruses like HIV directly attack immune cells or rapidly mutate to escape recognition. And in some cases, the immune system simply lacks the specific receptors needed to recognize particular threats effectively. These limitations have inspired scientists to ask a profound question. What if we could enhance, redirect, or reprogram the immune system to overcome its natural constraints? What if we could teach it to recognize threats it normally misses, to attack with greater precision, or to operate beyond its evolutionary programming? Emily Whitehead was just 6 years old when she became patient 1 in a clinical trial that would make medical history. After relapsing twice with aggressive leukemia and exhausting all standard treatments, her parents enrolled her in an experimental therapy at Children's Hospital of Philadelphia. Scientists would reprogram her own immune cells to hunt down and destroy her cancer. Initially, the treatment almost killed her as her reprogrammed immune cells triggered a massive inflammatory response. But when the storm passed, something remarkable happened. Her cancer completely disappeared. Today, more than a decade later, Emily remains cancer-free. Her case represents the first major success in reprogramming the human immune system to fight disease. The breakthrough that saved Emily's life is called chimeric antigen receptor (CAR) T-cell therapy, and it represents the first clinically approved method for reprogramming immune cells to target specific diseases. The approach is conceptually simple but technically sophisticated. T-cells are collected from a patient's blood, genetically modified in the laboratory to express synthetic receptors (CARs) that recognize specific molecules on cancer cells, expanded into the billions, and then rain-fused into the patient where they can seek and destroy cancer cells. These synthetic receptors are called chimeric because they combine components from different sources, typically an antibody-derived targeting domain that recognizes cancer cells fused to signaling domains from T-cell receptors that activate the cell when the target is found. Unlike natural T-cell receptors that require specific molecular presentations to recognize targets, CARs can be designed to bind directly to proteins on the cancer cell surface, bypassing some of the restrictions that limit natural immune responses. The first FDA-approved CAR T-cell therapies targeted CD19, a protein found on B cells and B-cell cancers like certain leukemias and lymphomas. The results have been remarkable. Complete remission rates of 60 to 90% in patients with relapsed or refractory B-cell malignancies that had resisted all other treatments. For many patients with previously incurable disease, CAR T-cell therapy has provided lasting remissions and the possibility of cure. However, the current generation of CAR T-cell therapies faces significant limitations. The treatment process is complex, expensive, and time-consuming, requiring cells to be shipped to specialized manufacturing facilities. The therapy can cause severe side effects, including cytokine release syndrome, a potentially life-threatening inflammatory response, and neurotoxicity. And while CAR T-cells have been highly effective against some blood cancers, they've shown limited success against solid tumors, which present additional challenges like physical barriers to T-cell infiltration and immunosuppressive microenvironments. Recent advances are beginning to address these limitations. Scientists are developing CARs with multiple targeting domains to reduce the risk of cancer escape through antigen loss. Armored CAR T-cells carry additional modifications to help them overcome immunosuppressive tumor environments. Inducible safety switches are being built into CAR designs to allow precise control of T-cell activity if dangerous side effects develop. Perhaps most excitingly, researchers are extending CAR technology beyond cancer. CAR T-cells have been engineered to target HIV-infected cells, suggesting potential applications against persistent viral infections. Early research is exploring CAR-modified regulatory T-cells to treat autoimmune diseases by suppressing harmful immune responses against specific tissues. The success of CAR T-cell therapy represents more than just a new cancer treatment. It provides proof of concept that the human immune system can be reprogrammed to recognize and fight diseases it would normally miss. It demonstrates that with the right engineering, immune cells can be given new targeting abilities while maintaining their sophisticated killing mechanisms, memory formation, and ability to proliferate in response to their targets. This first generation of immune reprogramming has already transformed our understanding of what's possible in medicine. But it may be just the beginning of a much broader revolution in how we treat and prevent disease. In 2022, scientists at the University of Pennsylvania announced a breakthrough that stunned the medical community. They had successfully used CRISPR gene editing to modify immune cells inside a patient's body, not in laboratory dishes but directly in living human tissue. This in vivo immune editing opens the possibility of reprogramming immune responses without removing cells from the body, potentially making immune therapy simpler, more accessible, and applicable to a much wider range of diseases. It represents just one of many frontiers expanding beyond the first-generation CAR T-cell approaches. While CAR T-cell therapy has demonstrated the powerful potential of immune reprogramming, it represents only the first step in what promises to be a rapidly evolving field. Multiple approaches are now being developed to enhance, redirect, or modify immune functions in increasingly sophisticated ways. T-cell receptor (TCR) engineering offers an alternative to CAR technology. Rather than creating synthetic receptors, this approach modifies the natural receptors that T-cells use to recognize targets. TCR-engineered T-cells can recognize proteins inside cancer cells, not just on their surface, potentially addressing a much wider range of cancer types. Companies like Adaptimmune and Eradicate are developing TCR-engineered T-cells for solid tumors that have proven resistant to CAR-T approaches. Natural killer NK cells provide another promising platform for immune engineering. These innate immune cells can naturally recognize and kill cancer cells without the same risk of triggering graft-versus-host disease, making them potentially suitable for off-the-shelf therapies derived from donors rather than individual patients. Researchers are developing CAR-NK cells that combine the targeting precision of CARs with the natural cancer-killing abilities of NK cells, potentially offering treatments that are more accessible and have fewer side effects. Macrophages, immune cells that normally engulf and digest pathogens or cellular debris, are also being engineered with CARs. CR macrophages could be particularly effective against solid tumors, as they naturally penetrate tissue barriers that T-cells struggle to cross. They might also stimulate broader immune responses against cancer by presenting tumor antigens to other immune cells after engulfing cancer cells. Beyond cell-based approaches, antibody engineering is creating new ways to redirect immune responses. Bispecific antibodies can simultaneously bind to cancer cells and T-cells, bringing them together to facilitate immune attack. Checkpoint inhibitor antibodies block signals that normally restrain immune responses, releasing the brakes on natural anti-cancer immunity. These approaches are already approved for multiple cancer types and represent a form of immune reprogramming that doesn't require genetic modification. mRNA technology, which gained worldwide attention through COVID-19 vaccines, offers another platform for immune reprogramming. By delivering instructions for cells to temporarily produce specific proteins, mRNA could potentially direct immune cells to express modified receptors or other molecules that enhance their disease-fighting capabilities without permanent genetic changes. Perhaps most revolutionary is the development of in vivo immune cell engineering, modifying immune cells directly inside the body rather than removing and reinfusing them. This approach could dramatically simplify immune therapies, making them more accessible globally and applicable to a wider range of diseases. Early clinical trials are using nanoparticles to deliver gene editing tools specifically to immune cells, allowing precise modifications in the body's natural environment. These diverse approaches share a common vision: harnessing the sophistication of the immune system while overcoming its natural limitations. Each technology offers different advantages and may be suited to different disease contexts. Together, they're expanding our conception of what's possible in medicine, suggesting a future where immune reprogramming could address virtually any disease that involves abnormal cells or foreign pathogens. As these technologies mature, they're likely to become more precise, more controllable, and more accessible. The current generation of immune therapies often represents a one-shot approach. Cells are modified once and then operate autonomously. Future systems might include sophisticated control mechanisms, allowing doctors to adjust immune activity in real time or deactivate engineered cells if necessary. This could expand applications beyond life-threatening diseases to chronic conditions that require ongoing management. In 2016, scientists at the J. Craig Venter Institute created the first synthetic bacterial organism with the smallest genome required for life. In 2019, another team at ETH Zurich built the world's first functional bacterial genome designed entirely by a computer algorithm. As synthetic biology advances from simple bacteria to more complex systems, a provocative question emerges. Could we someday design synthetic immune cells programmed to target any disease on demand? Could we create a universal platform for immune programming that adapts to new threats as quickly as software updates are pushed to smartphones? As we look beyond current technologies toward more speculative future possibilities, the concept of universal immune programming emerges, a hypothetical platform that could redirect immune responses against any disease target quickly, precisely, and safely. While entirely speculative, this concept builds upon technological trajectories already visible in today's research. A truly universal immune programming platform might include several key components. First, it would need a delivery system capable of reaching and modifying specific immune cell populations anywhere in the body. This could involve sophisticated nanoparticles designed to target particular cell types, synthetic viral vectors with precise tissue tropism, or even cell-penetrating peptides that carry modification machinery to specific immune cells. Second, it would require a programmable recognition system that could be rapidly updated to target new threats. This might build upon emerging technologies like DNA origami, which allows the creation of three-dimensional nanostructures with precise binding properties, or synthetic biology approaches that generate novel protein interfaces for molecular recognition. The system would need to incorporate safeguards preventing recognition of healthy tissues while maintaining high sensitivity for disease targets. Third, a universal platform would need tunable response mechanisms that could be adjusted based on the nature of the threat. Different diseases might require different immune approaches: direct killing for cancer or viral infections, phagocytosis for bacterial pathogens, or neutralizing antibody production for toxins. The ideal system would deploy the most appropriate response for each situation while avoiding excessive inflammation or collateral damage. Finally, it would incorporate control systems allowing precise regulation of immune activity. This might include molecular switches responsive to specific drugs, enabling doctors to modulate the intensity of immune responses or deactivate the system entirely if necessary. Temporal control mechanisms could ensure that engineered immune responses persist only as long as needed to address the threat. Several emerging technologies could contribute to this speculative future. Cell-free systems, biological machinery extracted from cells and reconstituted in synthetic environments, might allow rapid production of tailored immune components that could be injected and incorporated into the body's existing immune architecture. Synthetic genomics could potentially create artificial immune cells with capabilities beyond those found in nature, designed specifically for therapeutic purposes. Advanced biomaterials might serve as scaffolds for immune engineering, creating microenvironments that guide immune cell development and function in predetermined ways. One particularly intriguing possibility is the development of immune computers, biological systems that perform computational functions within the immune environment. These hypothetical constructs would sense multiple inputs, like molecular patterns associated with disease, process this information through biological circuits, and execute appropriate immune responses based on complex decision trees. They could potentially operate with sophistication similar to natural immune cells but with targeting and response parameters defined by human designers rather than evolutionary constraints. How might universal immune programming be implemented in practice? One scenario involves centralized response development with distributed delivery. When a new disease emerges, researchers would rapidly characterize its molecular signatures and design appropriate immune recognition elements. These designs would be synthesized and formulated into delivery systems distributed to healthcare providers globally. Patients would receive injections that temporarily reprogram specific immune populations to recognize and eliminate the new threat without permanently altering their genetic makeup. Another possibility is the establishment of permanent immune reprogramming platforms within individuals, perhaps cell populations engineered during early life that can be repeatedly updated with new targeting instructions as needed throughout life. These platforms might function as specialized immune apps that supplement rather than replace the natural immune system, providing targeted capabilities against specific disease categories. While these scenarios remain speculative, they represent extrapolations of current scientific trajectories rather than pure fantasy. The conceptual foundation for universal immune programming already exists in today's research, even if the technical implementation remains beyond our current capabilities. In 1980, the World Health Organization declared smallpox, a disease that killed an estimated 300 million people in the 20th century alone, completely eradicated through vaccination. It remains the only human disease ever fully eliminated. But what if advanced immune programming could do for all infectious diseases what vaccination did for smallpox? What if cancer became as treatable as a common infection? The implications would extend far beyond medicine, potentially transforming human lifespan, society, and even our evolutionary trajectory. If universal immune programming became reality, the implications would be profound and far-reaching, potentially transforming our relationship with disease and the trajectory of human health. Perhaps the most immediate impact would be on infectious disease management. When new pathogens emerge, whether natural outbreaks, mutated strains of existing diseases, or engineered biological threats, rapid immune reprogramming could provide immediate protection without waiting for traditional vaccine development. Pandemic prevention might shift from containment and emergency response to rapid immunological updating, with new immune targeting packages deployed globally within days of threat identification. For existing infectious diseases, the technology could offer paths to eradication currently considered impossible. HIV, tuberculosis, malaria, and other persistent infections might be systematically eliminated through coordinated immune programming campaigns. Diseases that have evolved sophisticated immune evasion mechanisms could be countered with equally sophisticated immune recognition systems designed to target their essential components. Cancer treatment would be transformed even more fundamentally than with current immunotherapies. Rather than the general approaches used today, treatment could involve comprehensive profiling of an individual's cancer followed by precise immune programming targeting its specific molecular signatures. As tumors evolve to escape initial targeting, the immune programming could be updated in real time to address new variants, creating an adaptive response that cancer cells couldn't outpace. Autoimmune diseases might be addressed through selective reprogramming of immune tolerance, teaching the immune system to recognize and accept specific self-tissues it had mistakenly targeted. Rather than broadly suppressing immune function as current treatments do, this approach could resolve autoimmune conditions while maintaining normal immune protection against infections and cancer. Beyond treating established diseases, immune programming could potentially address aging-related immune decline. As we age, our immune systems become less effective and sometimes dysregulated, contributing to increased disease susceptibility and chronic inflammation. Targeted immune rejuvenation might restore youthful immune function without disrupting the complex balance of immune regulation, potentially extending not just lifespan but health span, the period of life spent in good health. The cumulative effect of these capabilities could dramatically alter human longevity. While speculative, it's conceivable that effectively addressing the major causes of mortality through immune programming could extend average lifespans significantly. If cancer, infectious diseases, and cardiovascular conditions, which often have inflammatory components amenable to immune modulation, could all be effectively managed or prevented, the natural limits of human lifespan might be approached more commonly. These health transformations would have profound social and economic implications. Healthcare systems designed around managing chronic diseases might shift toward preventive immune monitoring and programming. The economic burden of disease could decrease dramatically, while productivity might increase as people remain healthy and active longer. However, extended lifespans would create new challenges for social systems like retirement planning and intergenerational wealth transfer. Global health inequities could either be exacerbated or reduced, depending on how the technology is developed and distributed. If immune programming technologies become widely accessible, they could help close health gaps between wealthy and developing nations more effectively than traditional medical approaches. Conversely, if access remains limited to wealthy countries or individuals, existing health disparities could worsen dramatically. Evolutionary implications also merit consideration. Throughout human history, infectious disease has been a powerful selective force shaping our genetic makeup. Widespread immune programming might alter these selective pressures, potentially changing the trajectory of human evolution in ways difficult to predict. Similarly, pathogen evolution might be affected as immune programming creates new selective pressures on disease-causing organisms. Perhaps most profoundly, advanced immune programming could change humanity's psychological relationship with disease and mortality. If previously inevitable diseases become readily manageable, our conception of the human condition, long defined in part by vulnerability to illness, might fundamentally shift. This could alter cultural, philosophical, and religious frameworks that have traditionally helped people make sense of suffering and death. While these implications may seem distant or speculative, they highlight the potentially transformative nature of the technology we're discussing. Immune reprogramming represents not just another medical advancement but a fundamentally new approach to human health that could reshape our species' relationship with disease. In 2015, scientists gathered at a historic conference in Washington, D.C. to discuss the implications of CRISPR gene editing technology. One presenter shocked the audience by revealing that his team had already edited human embryos, crossing a line many thought was still years away. The moment highlighted how rapidly biotechnology can advance and how ethical frameworks often struggle to keep pace. As we contemplate the potentially revolutionary impact of immune reprogramming, we must also honestly confront the profound challenges, risks, and ethical questions this technology would present. Despite its extraordinary potential, universal immune programming would face substantial technical challenges and raise serious ethical questions that demand careful consideration. The technical challenges are formidable. The immune system's complexity makes precise reprogramming extraordinarily difficult. Unintended consequences could emerge from modifying such an interconnected system, potentially triggering autoimmune reactions, excessive inflammatory responses, or immune dysregulation that creates new vulnerabilities. The balance between effective immune response and dangerous overreaction is delicate, and disrupting this balance could have serious consequences. Safety concerns are paramount. Any technology capable of reprogramming immune responses carries inherent risks of triggering cytokine release syndrome, a potentially fatal inflammatory condition seen in some CAR T-cell patients. Long-term effects of immune modification remain largely unknown, with potential risks including chronic inflammation, immune exhaustion, or increased susceptibility to certain pathogens. Extensive safety testing would be essential before widespread implementation. Dual-use concerns also merit serious attention. Technologies developed for therapeutic immune programming could potentially be misused to create biological weapons targeting specific populations based on genetic markers. The democratization of biotechnology makes controlling such dual-use risks increasingly difficult, requiring international governance frameworks that balance innovation with security. Privacy issues would emerge around the genetic and immunological data required for personalized immune programming. This information could reveal sensitive details about individual health predispositions and might be valuable to insurers, employers, or other entities. Strong privacy protections would be essential to prevent discrimination or exploitation based on this data. Access and equity considerations are equally important. Initial versions of immune programming technology would likely be expensive and available primarily in wealthy countries or to affluent individuals. Without deliberate efforts to ensure global access, the technology could dramatically widen health disparities. Developing deployment models that prioritize global health equity from the outset would be essential for realizing the technology's full humanitarian potential. Regulatory challenges would be substantial. Current regulatory frameworks for medical technologies were not designed for therapies that might fundamentally reprogram biological systems. New approaches would be needed to assess safety, efficacy, and long-term impacts while enabling innovation. International harmonization of these frameworks would be crucial for addressing global health threats effectively. Perhaps most profound are the philosophical and ethical questions surrounding human intervention in fundamental biological processes. How much should we alter systems shaped by millions of years of evolution? What unintended consequences might emerge from redirecting immune functions beyond their evolutionary contexts? These questions have no simple answers but require thoughtful engagement from diverse perspectives. Religious and cultural considerations will also influence societal responses to immune reprogramming. Many traditions have specific views on medical intervention, modification of natural processes, and the proper limits of human control over life and death. Respectful dialogue across these traditions would be essential for developing widely acceptable approaches to implementing these technologies. Despite these challenges, the potential benefits of immune reprogramming may justify careful development with appropriate safeguards. The history of medicine includes many innovations that initially raised serious concerns but ultimately became standard care because their benefits outweighed their risks when properly managed. The key lies in thoughtful, inclusive approaches to technology governance that consider diverse perspectives and prioritize both innovation and safety. Rather than viewing these challenges as reasons to avoid developing immune reprogramming technologies, we might better see them as a call for responsible innovation, developing these powerful capabilities while simultaneously building the technical safeguards, ethical frameworks, and governance systems needed to ensure they benefit humanity broadly. The future of immune reprogramming will be shaped not just by what becomes technically possible, but by the choices we make about how to develop and deploy these technologies. With thoughtful approaches that prioritize safety, equity, and ethical considerations alongside innovation, immune reprogramming could represent one of humanity's most significant advances in addressing disease and promoting health. As we stand at this frontier, the question is not simply whether we can reprogram the immune system to target any disease, but how we can do so in ways that reflect our highest values and aspirations as a species.